Great. So I'm going to talk to you about T-cell therapy, kind of current applications and future directions, and tell you a little bit about the field, what CAR T-cells are, and where we're going both in the laboratory and in the clinic. So just as disclosures, all the therapies that I'm going to talk about are investigational. No CAR T-cell or T-cell therapy is currently approved by the FDA for anything. I'm also wildly conflicted in that I consult for various companies that have sprouted out around cell therapies, and I do hold some I am an inventor on some patents that came out of Penn. So as you may know, cancer immunotherapy has moved really from the fringes to the forefront of immunology and oncology. 10 years ago when we were giving these talks, they were pretty empty, small rooms in the corners of SITC and ASGCT, these very small meetings. Now the conferences have exploded, the rooms are packed, and so it's really, really different. And it's because it's working in the clinical setting. And even though immunotherapy had offered this possibility of curing cancer for hundreds of years, it was really hard to do. And the reason for that is because the immune system has really evolved to be tolerant to self-tissues. And most of the tissues that are, most of the antigens and proteins that are expressed in tumors are, of course, derived from self. And so it's very difficult to get a T-cell to recognize a tumor. And so what's really changed in the last 5 to 6 years is that we've now figured out ways to block the two main forms of tolerance. So the first main form of tolerance to block is peripheral tolerance, and this is where the tumor actually has immunosuppressive functions against the T cell. So these are molecules like PD-L1, for example, that block T cell activation. So this kind of assumes that the T cell will recognize antigens on the tumor in the context of MHC and peptide. And the T cells are there and they can recognize particularly these neoantigens that you're going to hear about from Kathy Wu after me. But because the tumor is secreting these immunosuppressive molecules or ligands, it blocks the T cells. And this is a form of peripheral tolerance that the tumors usurp for the express purpose of blocking the immune system against them. The other form of tolerance, though, is central tolerance. And this means that the T cells that live in our bodies normally are deleted when they recognize anything that's derived from self. And so as they're growing up in the thymus and they're getting educated, most of the T cells will not recognize anything on the tumor cell. And so what we do now is we can genetically modify the T cells with designer antigen receptors. So we can basically put a GPS on them and tell them, go find the tumor, and then the T cell knows what to do. It'll kill it. Once it engages its target, it'll kill. But basically, most of our T cells are not very good at finding the tumors. And so now we can use genetic engineering to try to engineer the T cell receptors or chimeric antigen receptors into T cells to tell them where to go. And so there's basically 3 components to T-cell engineering. This is something that's been developed over about 20 years. One is using a gene delivery system. We've actually co-opted what we've learned from viruses, in particular HIV, to try to transduce T-cells, meaning use the vector to insert the new DNA or the new nucleic acid material to code for the new antigen receptor. We've had a lot of fun also designing antigen receptors. So one can use either natural T-cell receptors, which are actually fairly complex molecules because they're heterodimers and they associate with other signaling molecules, or one can make completely engineered kind of antigen receptors like chimeric antigen receptors or T-bodies that take portions of an antibody and fuse them to the signaling domains of T cells. And the third important component of engineering T cells is a way of being able to grow the T cells while they're outside the body. So usually when you take T cells out, you have to be able to stimulate them, keep them in culture, get them to the target cell dose, and then infuse them back into the patient. And if you don't stimulate the T cells well while they're outside the body, then when they go back in, they just kind of disappear and die off. So you really have to have a mechanism to grow them while they're outside the body so that they retain the 2 most important T cell properties. And I'm very T cell focused, and you'll see why in a few minutes. But the most important T cell properties are the ability to kill upon target recognition and the ability to form long-term memory. So once they see the antigen once, they're primed to kill. They see it twice, and then they actually kill much faster and much stronger. And they also divide and repopulate so that they can keep killing more cancer. So there's 2 kinds of antigen receptors that have been described. And it's important because they actually encompass all the ways that one could potentially see antigen if one is a T cell. So one is this chimeric antigen receptor, which I introduced briefly. This is based on an antibody, and so it recognizes what's on the surface of a cell in its native conformation. The alternative is a T-cell receptor, which recognizes bits and pieces of processed protein that are presented on the MHC molecules. So really, CARs only recognize what's on the surface of cells, while T-cell receptors can recognize almost the entire array of proteins that are in the cell. But it's limited by this MHC expression. And this is unique to most individuals. There are some overlapping HLA molecules, but this is the most polymorphic region in the human genome. So designing T-cell receptors, you end up limiting your population to patients who have that particular MHC. The advantage of CARs is that you can make, with one vector, you can make them recognize any particular target regardless of MHC restriction. So the way this therapy works is a little more complicated than traditional chemotherapy or biologic therapy drugs because we're administering cells. And in the vast majority of cases now, we're administering autologous cells, meaning the same patient's immune system is engineered to recognize their cancer, and then those same T cells have to be gotten back to the patient. That's a pretty significant logistical endeavor. And so what happens is that the patients need to go through the blood bank and get a leukapheresis, which is a procedure where their white blood cells are collected. This product is then labeled and shipped to a central processing facility, which is what happens down here in yellow. The T cells are stimulated to make them grow. The gene is introduced using a lentiviral vector or a retroviral vector. They're expanded. And then all of the excipients are removed, so things like anything that you added to the culture, beads, et cetera, needs to get washed off and removed. The cells are processed, frozen, tested for lack of contamination with any bacteria and things like that before they're given back to the patient. And then they're shipped back to the patient where they can be infused at the bedside. So some of the complex logistics here involve the manufacturing process, the chain of custody, and then the infusion. We're thawing cells at the bedside or thawing cells in a specialized laboratory and bringing them to the patient. It's just a very different model than what is traditionally given in cancer therapy. But it's incredibly powerful. So this is an example of a T-cell receptor modified T-cell. These T-cells are recognizing the cancer testis antigen NY-ESO-1. And this is a trial that's sponsored by Adaptimmune in patients with synovial cell sarcoma who have the particular MHC called HLA-A2 and who express the NY-ESO antigen in their tumors. And what you can see is that there are these complete responders when patients have metastatic disease to the lung here. There's an initial, actually, pseudoprogression when the T cells catalyze a inflammatory infiltrate, and it can initially look worse on a CT scan, but then these patients can go into complete remission. And these are still small studies, so this is just 10 patients, but there's a 60% response rate in this particular disease that is otherwise refractory to other therapies. Now, that was a T-cell receptor. What about these chimeric antigen receptors? Well, there's been a lot of hype and also opportunity in this field. So the first target that was really developed is CD19. And CD19 is a particularly good target because it is expressed in a wide variety of tumors, all derived from B cells. So everything from pre-B cell ALL to chronic lymphocytic leukemia, multiple myeloma, mantle cell lymphoma, all of the lymphomas pretty much. And it's not expressed on any normal, healthy, life-sustaining tissues. It is expressed on normal B cells, but you can live without your B cells. And it's not expressed on anything that is absolutely required to have a functional and happy life. And so one can target CD19 knowing that the T cells transduced with the CARs will be able to recognize anything that expresses CD19. And what's amazing is that this actually worked. And it is working, and it's working for a variety of reasons. So the first times that people made CARs, they put in just a single intracellular signaling domain, but then those actually didn't work, and those were tested about 15 years ago. But we know a lot about T-cell activation, and that T-cells require multiple signals to be able to form long-term memory and to be able to have adequate stimulation to be able to go on and multiply and keep killing tumor. And so different stimulatory molecules were tried to put on the inside portion of the CAR, and what's really worked are the ones that are based on either CD28 or 4-1BB. And there's a lot of different co-stimulatory molecules that could be tried. And so as a platform, it's very appealing because one can engineer T-cell receptors and chimeric antigen receptors in different ways to make the inside portion of the T-cell do whatever we want it to do, either turn off or turn on, secrete cytokines or not secrete cytokines. So we can really have pretty exquisite control over the effector functions of the T-cells. So these are the three chimeric antigen receptor T-cells that are in clinical trials in the most advanced forms right now. These are all directed to CD19, and I just have this drawing here that was developed by Marco Davila that just shows how these differ. And so the one that was developed at Sloan Kettering, which is now called JCAR015, that's licensed and owned by Juno, has a single chain variable fragment, which is the antibody portion that recognizes CD19, and it has a CD28 signaling domain and the T-cell receptor signaling domain to make T-cells kill. The NCI had also developed a separate chimeric antigen receptor, which is very similar to the Sloan Kettering one, except that it uses a different antibody binding portion. And Seattle and Penn developed a third variation on this kind of chimeric antigen receptor, where the intracellular signaling domain differs, and it's 4-1BB. Now, they differ not only in how the receptors are designed, but also how the cells are cultured and processed, and how the CAR is introduced into the T cells. So JCAR015 uses a retroviral vector, and it uses a bead stimulation to grow the cells. These CARs last about 30 days. Kite uses also a retroviral vector, but they use a different way of growing their T cells. And Juno uses a lentiviral vector, and Novartis uses a lentiviral vector, but they both use different ways of growing their T cells. These particular CARs have been shown to last more than 4 years, and now 5 years in some of these patients. So the first 3 patients that were treated at Penn were treated with the last CAR that I showed you, which had this 4-1BB signaling domain. And the responses were pretty amazing. So these are all patients with relapsed and refractory chronic lymphocytic leukemia. And in this disease, there's 3 compartments of disease. One is in the peripheral blood, which is shown here, the bone marrow, and then the lymph nodes. And what you can see in the first 3 patients is that there were complete responses in all of the compartments for all of the patients. So in this case, we're looking at peripheral blood, where there's large tumor burden. Shown here, the patient gets a single infusion of CAR T cells, and the tumor burden decreases rapidly. This was associated with a flu-like syndrome, though. The patient was started on steroids, and eventually their disease returned. In this patient, you can see that 6 months after a single infusion of CAR T cells, there's no more B cells in the marrow, and also no more CLL. And in this patient who had this axillary lymphadenopathy, you can see that the lymphadenopathy has disappeared by 3 months, and this patient also went on to have a complete remission, which lasted now more than 5 years. So this is a pretty powerful non-cross-resistant form of therapy compared to biologic therapy like anti-CD20, Rituxan, and compared to chemotherapy. It turned out that what seems to be the greatest predictor of response is how these T cells work when they get back into the body. So unlike chemotherapy, where the PK is highest within a few hours of dosing of the drug, the T cells are living drugs. And so the dose that you infuse actually has to replicate inside the patient in order for it to work. And this became evident in the course of these initial trials, where these are the first 14 patients treated with CLL. And what you can see is that the complete responders have the highest levels of T-cell expansion. And the non-responders really do not have this T-cell expansion. And so that was really the first correlation between T-cell expansion in the body and response. And not only do they have this high expansion, they also tend to have this high persistence, meaning that the T-cells last for a year in many of these patients, and some of them they last more than 5 years. Whereas the patients who have even an initial expansion, but then they go away, they may have a partial response, but they tend not to have a complete response. Now that's true for this disease with this CAR. If you look at some of the data coming from KITE, which is going to be presented at ASH in a month or so, this may not necessarily be required for every CAR in every disease. So they have a different model of a CAR, and they're mainly testing in diffuse large B-cell lymphoma, and it may be that these long-term persistence and this initial huge expansion is not required for every CAR. So unfortunately, although there is some overlap, you can't completely extrapolate data from one CAR in one disease to a different CAR with a different disease. Now, what about acute lymphoblastic leukemia? Well, CLL was good, but ALL was amazing. So the first patients treated with pre-B-cell ALL that was CD19 positive had these very, very rapid inductions of remission and long-term complete remissions. So just as an example, this is the first pediatric patient treated with CD19 CAR T cells. And you can see here in the marrow, this is the CAR T cell expansion. And at day 6, you see this small proportion of T cells that are actually CAR positive. And 16 days later, they've had this massive expansion where 3/4 of the T cells in the patient are actually CAR T cells. And coincident with this, you see that the blasts, the ALL blasts, go away. And this patient is now in MRD negative complete remission for 4.5 years. if we look at the patients, the first 30 patients that were treated with B-cell ALL, these are also pretty remarkable. So this was published almost 2 years ago now in the New England Journal of Medicine. In the first 30 patients treated at Penn, there's a 6-month overall survival of 78%. In patients with relapsed refractory leukemia who would otherwise have survival curves that look like this. So these are all patients who had had multiply relapsed leukemia. Several of them had already had an allogeneic bone marrow transplant, and there were really no other choices. Now, there's going to be data coming out that show that this is true not only for CD19, but can also be true for other CARs targeting other antigens in leukemia. And this can actually work not only with this particular CAR, but with many other different variations of CARs with different signaling domains and different ways of growing the T cells. So as a platform, it's broadly applicable to multiple diseases. Most of the data has been in B cells, so CD19 is very promising, CD22 is looking very promising, BCMA for multiple myeloma is looking very promising. There hasn't been anything in solid tumor yet that has really worked in the setting of a CAR. The best has been the T-cell receptors that I showed you with the synovial cell sarcoma. So you can see here that multiple different groups have had these amazing complete remissions. This was the talk of ASCO last year. It's going to be the major talk of ASH this year as well. So the groups from Seattle are seeing 94% complete remissions in adults and in pediatrics. The groups from Penn in adults have a slightly lower rate of complete remission, although they also did not use fludarabine in their lymphodepleting chemotherapy. And in pediatrics, very high rates of remission. In adults, a little bit lower. And that's partly associated because the toxicities in adults tend to be worse. And so we use lower doses and intervene faster with steroids and toxicity management. So the biggest issue that has actually emerged in the complete responders is the emergence of resistance. And this can happen in 2 different ways in ALL, actually 3 different ways now. So one is if you lose, if the CAR T cells have this initial expansion but then don't persist, then the B cells come back and the tumors come back. And we think that we need at least 6 months of persistence in order for the T cells to have a complete remission to be effective. So about 33% of the relapses are actually CD19 positive. And in those cases, some of the patients have had a second infusion of a CD19 CAR, and they can go back into remission. Now, there are some issues about giving the same CAR back to a patient because they can actually develop immune responses to the CAR. And so it's possible that having a humanized CAR or a different version of a CAR could be better as a second infusion rather than administering the same one again. Unfortunately, the more common form of relapse, which occurs in about 2/3 of the patients, and about 30% of them relapse overall in ALL, is with CD19 negative disease. So the cells are actually losing the target. And there's been a couple of different ways that have been described for how the cells do this mechanistically. One is that they actually just lose portions of the CD19 that are recognized by the CAR, and this can occur through different splice variants. So this is what the CD19 molecule looks like. It has these 3 sort of immunoglobulin-like domains, transmembrane domain, and then a signaling domain. And all of the CAR epitopes and antibodies recognize this portion, which is exon 2. And so the 2 splice variants that have been described, for example, just lose the exon 2 but keep the rest of the molecule, or lose the transmembrane domain so that it's not anchored to the tumor anymore. And this is actually retained sometimes in the tumor cell so that they can still have the signaling domains of the CD19 that are active. And so that kind of implies that CD19, even though it's not an absolute driver or required for the tumor phenotype, it does provide some survival advantages because the cells like to maintain the CD19 intracellular portions. The other way that's been described to lose CD19 expression, and this is only in ALL, is a reversion to a myeloid phenotype. So the cells kind of go back into their stems, means the mutations are back in the hematopoietic stem cell, and then they redifferentiate and express all the myeloid antigens and not the lymphoid antigens anymore. And that's been described in a series of cases. And I suspect that there will be more. Now, this is how ALL relapses. It's not clear that the other diseases that are being targeted with CD19 CARs will relapse in this fashion. So there's only been one description of a CD19 negative relapse for CLL, and it's less likely, I think, to occur through this dedifferentiation and myeloid differentiation, again, just because CLL is a much more mature B cell at the time that it starts to become tumorigenic. So overall, the response rates in CLL and in lymphoma are a little bit lower than ALL, more in the 30 to 50% range. But still, in patients with relapse and refractory disease, this is something that is being pursued as a potential indication by multiple companies, including Kite and Juno, and eventually Novartis as well. So what about the toxicities with CAR-19? The main toxicity with CAR-19 is cytokine release syndrome. When there's a lot of T-cell activation and a lot of tumor cell killing, the T-cells are very bioactive. They make cytokines that make interferon gamma. They activate the myeloid compartment to make interleukin-6. And clinically, the patients develop fever, hypotension, a sepsis-like picture, capillary leak. They have multiple laboratory abnormalities. C-reactive protein comes up early and is associated with IL-6 because it's downstream of the IL-6 signaling pathway. There's also acute phase reactants like ferritin, and there can be organ damage. Severe cytokine release syndrome is very much like macrophage activation syndrome. There's organomegaly, hypercoagulability, and it's associated with high levels of circulating interleukin-6 and interferon gamma. It's managed primarily with anti-cytokine therapy, and that's usually the go-to management drug as opposed to something like corticosteroids, because steroids can actually decrease the initial expansion and function of the T cells. And so in an effort to avoid killing the active drug, we try to use anti-cytokine therapy. However, neurologic toxicity is also quite common. This is characterized by aphasia, can be seizures, can be multiple different forms of encephalopathy. Most often is quite transient, and it doesn't seem to respond to anti-IL-6 therapy. So it's really quite different. And so there's a balance between managing cytokine release syndrome with anti-cytokine therapy and managing neurotoxicity with more steroid type therapy, because the anti-cytokine doesn't really help. The final toxicity is actually the most common and most expected with any kind of T-cell therapy, which is on-target toxicity. So as I said, T-cells can recognize very low levels of antigen, and they don't discriminate between a tumor and a normal cell. If the antigen is there, it's a potential target. And so one of the expected toxicities with CD19 is a B-cell aplasia, which fortunately can be managed with infusions of IVIG or even now sub-Q infusions of IVIG. I think one of the most interesting and powerful indications of T-cell therapy is that really the only toxicity is related to on-target toxicity. If we think about cytokine release syndrome, it's because the T-cells are active. So it's not like chemotherapy when you lose your hair because the chemotherapy happens to react with hair follicles. If you're getting cytokine release syndrome, it's because there's so much tumor killing going on. And so there's a much higher tolerance for that occurring, I think, than sort of random organ damage that isn't actually directly related to the tumor effect. But this also makes defining the targets to use against the cancer for a T-cell therapy much more important. So these are not all from one T-cell therapy. These are all different kinds of T-cell therapy, and I'll just show them to you quickly. So when you direct T-cell receptors to melanoma site antigens or melanoma-associated antigens, you can get pretty significant rashes. If you do the melanocyte differentiation antigens, they can also cause uveitis. They can cause infiltration into melanocytes. If you target CEA with a T-cell receptor, you can get pretty significant colitis. If you target antigens that are also co-expressed in the brain, you can get pretty significant neurologic toxicity as well. And so really the main toxicity for the power of T-cells is that they do recognize everything on target. And finding the right targets, therefore, becomes the main challenge in identifying new kinds of T-cell therapies. So there's a couple of rare and theoretical toxicities which I just want to briefly mention, but they occur so rarely that I think, for the most part, these are not going to be game changers. So one is this on-target toxicity, which is the game changer, which I told you about. The other is off-target toxicity, so when a T-cell recognizes an antigen that's not what it's meant to recognize. This doesn't really occur unless you don't know really what the specificity of your antigen is. There's also systemic cytokine release syndrome. One can develop an allergy to portions of the CAR. So if you get multiple stimulations, you can also become hypersensitive to the genetically modified product. And then there's the three that really dictate a lot of what we do in terms of the regulatory reporting is for integration site oncogenesis. So we need to follow the patients for 15 years to know that what we integrated into their T-cell genome is not going to cause a leukemia. And there's the possibility of generating replication-competent vector as part of the manufacturing process. These two theoretical toxicities actually, I think, are what drive a lot of the cost and complexity of these therapies, but they've never actually been demonstrated. And so I'm hopeful that as we get more comfortable with these cells and as they become part of the standard medical care, that we'll continue to be vigilant for it, but hopefully not quite, it will not be as burdensome. So the current landscape in CAR T cells is that for CD19, there's actually 3 potential indications that will be happening in the next year or so, in lymphoma, in leukemia, and in ALL. So CLL, non-Hodgkin's lymphoma, and ALL. There's also potentially CD22 that is a little behind CD19, but it seems to work even in CD19 negative patients, and so that could be a backup. And now, of course, there's going to be the combination of administering 2 at once to try to prevent antigen escape. There's also ongoing studies in targeting B-cell maturation antigen, which is expressed later in B-cell development and is frequently expressed in multiple myeloma. And there's still very early stage trials addressing using CARs in other hematologic malignancies. These are all still more academic trials. So people are targeting AML with CD33 and CD123. People are targeting Hodgkin's disease with CD30. People are using combinations of 19 and 123 for various kinds of leukemias as well. So I think there's going to be a lot coming out, particularly in the hematologic malignancy space, where we know that this can be a highly effective therapy. So I'll tell you just a little bit about BCMA. BCMA is an antigen that's expressed late in B-cell development, and it's expressed in about 90% of multiple myeloma cells. And there's actually already been a trial published by Jim Kokendorfer from the NCI using a BCMA CAR that has a CD28 signaling domain. He treated 12 patients in a phase 1 study, had 4 responders, but really started at quite a low dose. And the higher doses are the ones that had responses. And this has now been, I don't know exactly if it's been licensed or what the relationship is, but this is being further developed by Bluebird Bio now. They have changed the construct of the CAR and changed some of the manufacturing process, but they have an ongoing study that's open here and multiple other places using BCMA in patients with relapsed and refractory multiple myeloma. And we're seeing the same sorts of toxicities that one would expect for CD19, meaning on-target toxicity towards plasma cells and some cytokine release syndrome. So what about solid tumors? For solid tumors, there's a couple more hurdles that we don't have to deal with as much in the hematologic malignancy space. One is targeting specific antigens. We don't know as much about what is expressed exclusively in a solid tumor on the surface of the cell compared to a hematologic malignancy. We also don't know exactly what the rest of the landscape looks like for any particular target in the body. And so figuring out a way in a preclinical model or with human tissue to figure out where that target is and to make sure that it's on a dispensable tissue is going to be important. Some of the other factors are sometimes solid tumors have a pretty extensive tumor stroma, and it can be hard to know or even to measure about whether the T cells are able to cross into that, either due to physical barriers or biological barriers, where the T cells have either chemokines that don't let them get in, or the tumors secrete molecules that block the T cells biologically, even if they do penetrate. The other question, of course, is what the effect of the tumor microenvironment is going to be. So even if the T cell penetrates the tumor stroma, gets to the tumor, will it be completely inhibited by the same molecules that inhibit normal T cells infiltrating tumor? So things like PD-L1, et cetera. And we do know that CARs are susceptible to some of those inhibitory molecules, but they seem to be less susceptible than non-modified T cells. And it seems to me like one could engineer the signaling domain so that they become resistant to it, or one can genetically modify the T cell to become resistant to those inhibitory molecules. So there's ways around this, but they all need to be figured out, I think, for solid tumors. There's multiple solid tumor CAR targets that are in development. I don't want to go through the whole list, but I just wanted to highlight EGFRV3 because that's the one I've been working on for the last 2 years, so I can show you some data about it. EGFRV3 is a molecule that's it's a mutation in the epidermal growth factor receptor in the extracellular domain. So there's deletions of exons 2 through 7, which is a big chunk of the extracellular domain of EGFR. And there's this insertion of this new amino acid here, a new glycine. And so one can make an antibody to it. And because it's on the surface of the cell, it becomes fairly straightforward to turn that into a CAR. The trouble with EGFRV3 as a potential target antigen, it really stems from 2 factors. One is that it's heterogeneously expressed. And so even in patients who have EGFRV3 positive tumors, it tends to be a small portion of the cells that are EGFRV3 positive. And so there's already going into it the expectation that there's going to be some antigen escape. And the other is that it's expressed mainly in glioblastoma and only in a fraction of glioblastoma patients, about 20% of them. And so it's a relatively small population. Getting T cells to traffic into the brain is not necessarily trivial. And so developing a new CAR to a glioblastoma antigen can be quite challenging. We did open a clinic. We did, though, develop a new CAR directed to EGFR variant 3, and after extensive in vitro studies and mouse studies showing that this worked beautifully in the mouse, we opened a phase 1 clinical trial and treated 10 patients. The first thing we wanted to look at is to see whether we had that same level of T-cell expansion in the blood that we see with the CD19 CARs. We didn't expect it to be the same levels because it's a much lower tumor burden. We're talking about a gram of tumor just in very high-priced real estate in the brain compared to kilograms of tumor in the blood and the circulation. And we also knew, we knew that there would be low levels of antigen, and so we didn't expect there to be these high engraftment. But we were able to detect engraftment, and by engraftment I mean specifically when you infuse the cells and then you take a peripheral blood sample, can you find them again? And in all of the patients we were able to find them again, and at reasonable levels, but about 50 to 100 times lower than what we saw with CD19. We also didn't see this long-term persistence. We didn't see anybody who had large numbers of T cells detectable after day 28, and we think there could be multiple reasons for that. One is that they're not seeing enough antigen, and so you're not getting expansion. The other is that they're all going into the brain. That would be the best potential outcome, and they're not hanging around in the blood where there isn't really any antigen. And the other is just that there's T-cell homeostasis, that you don't maintain large portions of antigen-specific T cells in any one particular compartment. So we don't really know the reasons for that, but those were the observations. What we did find is that in at least a couple of patients, the T cells do seem to be going into the brain tumor and actually expanding there. And so this is looking at CAR T cell numbers in the blood versus the brain. And so if you saw the same level, it would be a ratio of 1 in the blood and the brain. But in at least 2 of the patients, there were actually many more T cells in the brain tumor than in the blood. And so we do find that we can find T cells that are trafficking into the brain and that they're expanding there. And coincident with this, most of the patients also had decreased levels of antigen expression in pre-CAR T-cell biopsies compared to post-CAR T-cell biopsies. Now, getting biopsies in patients with glioblastoma is not a trivial thing. So this was done partly for clinical reasons, and not everyone had their biopsies at the same time. So it's not the most controlled study in that sense, but we were able to get as much information as we could from the samples that we were able to obtain for clinical need. So we are able to see that there's CAR T-cell infiltration into the brain and anti-target activity. What we also saw, though, is that there's a lot more T cells just getting into the brain after CAR T-cell infusion than there were before CAR T-cell infusion. So this is sequencing all of the T-cell receptor beta chains. This is using the adaptive technology. And in the green boxes are the number of unique T-cell receptor chains that were present in the brain tumor before CAR T-cell infusion. So it's actually the green boxes, I guess, plus the blue, which are shared. And what's in the red boxes are the number of unique T-cell receptors that are found in the tumor after infusion. And what you see is that in each case, there's about a 20-fold increase in the number of unique TCRs that are in the brain tumor. And so at first, we were really excited thinking we're getting epitope spreading, which means that the T cells initially started an immune response to the tumor and then released a bunch of antigen, and we were able to stimulate new T cells coming in to recognize those antigens. Unfortunately, though, that didn't seem to be what was actually happening. So we were able to develop an assay with Novartis to look specifically for the phenotype of the infiltrating T cells into the FFPE tumor blocks that we had. And so we saw that there was this huge T cell infiltrate. This is the pre-CAR T-cell infusion. This is the post-CAR T-cell infusion. And what you can see here is this is the CD3 infiltrate in the pre-CAR T-cell infusion, just small numbers of T cells going in. This is the post-CAR T-cell infusion with these very large numbers of T cells going in. Some of these are CAR-specific, but many of them are not. So this is a specific probe to look for CAR sequences. And you can see that there's occasional CAR-positive T cells here, but most of them are actually not CAR-positive. And they're not really making interferon gamma, and some of them may be making some granzyme B, and these are the 2 molecules that are most associated with T-cell killing function. There's a reasonable number of CD8s compared to CD4s, And many of them are also positive for CD25. And CD25 is interesting, like many of the other T-cell activation molecules, and that it signifies that a T-cell has recently seen antigen, but it's also, so it's called an activation marker, but it's also expressed on all of the regulatory cells, so the cells that actually inhibit T-cells. And so what we found looking further is that many of these T-cells are actually the inhibitory regulatory T-cells. And so we see that they express high levels of PD-L1, high levels of FOXP3, and the tumor has high levels of IDO, which is also an inhibitory towards T-cell function. And this is actually higher than they were in the pre-CAR T-cell samples. So what we think happened is that the T-cells got in, they got activated, they secreted even a little bit of interferon gamma, which then recruited all of these adaptive resistance pathways and brought in new Tregs and immune inhibitory markers. In this particular patient, there weren't high levels of TDO, IL-10, or TGF-beta, but when we look at all 10 patients where we had data, all of them upregulated one or more of the inhibitory pathways towards T cells. So the good news is that we were able to get T cells that were safe, that engrafted, that got into the brain, but the bad news is that they encountered a very, very inhibitory microenvironment, and they didn't stick around long enough to really have long-term effects the way that we have been seeing with CD19. And so there's a lot of next-generation CAR designs besides just targeting a single antigen, which we think is going to be a major, major barrier to extending this platform technology for most solid tumors. And so there's a lot of interesting things one can do with T-cell engineering. We can make bispecific CARs to recognize two antigens, and you can put logic gates into them. So you can put two antigens to avoid antigen escape, like in the context of CD19 and 22, to try to avoid 19 or 22 escape. So using an OR gate, or you can design CARs to avoid toxicity so that a CAR or a T cell has to recognize two antigens and only go when both antigens are on the tumor. And so those are all still in the preclinical setting and getting published in the scientific journals. There's also alternative intracellular signaling domains that are being described. I showed you a slide early on that showed all of the different molecules that can direct T-cell fate, and so I think there's a lot of systematic investigation now of what happens when you put each of those into a CAR. Can you make T-cells turn on and off in response to different stimuli and change T-cell fate? There's also alternatives to the extracellular portion of the CAR. So besides using a standard murine-based antibody, people are using antibody-coupled receptors. They're using DARPins. They're using camelid CARs. And so there's a lot of different ways that one can imagine protein-protein interactions becoming the extracellular portion of an engineered T-cell. There's also an effort to have drug-controlled CARs, so CARs that have moieties that dimerize only in the presence or absence of a drug. And so that would make a CAR a physician-controlled drug where the patient can theoretically get the infusion once, and then if you administer a drug, the CAR turns on, or the opposite, if you administer the drug, the T-cell commits suicide. And so this has some appeal to be able to have the physician control what happens to the CAR T-cell product once it's back into the patient. Right now, we put it in, but then we don't really have a lot of control over what happens to it afterwards. They expand or they shrink kind of outside of physician control, but under biologic control. There's also a huge effort to develop allogeneic CARs. So this would get around all of the complexity of the chain of custody and manufacturing of using a patient's own T cells. There are a lot of biologic barriers to overcome with this. Right now, we do full HLA matching for bone marrow transplant as much as we can, and controlling GVHD and controlling T cell biology is actually quite challenging. But now with genetic engineering strategies, we may be able to genetically modify a product from one patient's T cells or from one normal donor T cells and then put them into the patient so that they can have the killing effects, but then not have graft versus host disease and not be rejected by the donor. So there's a lot of ongoing investigation to try to make allogeneic CARs. I think this is very appealing to the industry because then they have a drug product that can be sold, as opposed to having this complex chain of logistics where it's not really clear what the product is, and there's a lot of sort of anxiety about that. There's also other kinds of antigen receptors based on either T-cell receptors or their variants, which are in development by a lot of companies that are growing up around here. So our lab is working on turning immune cells into cancer fighters, and we're focusing on 3 different things, and each of them has 2 subgroups. So one is to improve the safety for hematologic malignancies. So we think that we can make T cells have less cytokine release, but still being able to maintain their killing function and still be able to have their long-term persistence. And we think that we'll be able to control trafficking. So we don't think, for example, that we need BCMA-positive T cells to get into the CNS because myeloma doesn't typically reside in the CNS. And so we may be able to avoid neurotoxicity by controlling T cell trafficking. Another thing is to improve the potency for solid tumors. So for solid tumors right now, we're seeing a lot of safety. We're not seeing a lot of potency. And I think that's for two main reasons. One is the heterogeneity of the antigen expression in any particular tumor, and the other is the inhibitory microenvironment. We've observed both of those things in our GBM trial, and I think it's been hypothesized for many other CAR T-cell trials in solid tumors, although it has not really been formally shown except for the GBM study. We also want to improve the feasibility for all kinds of CAR T-cells, and I think that that will enable us to go after new targets, to alter the manufacturing process so that it's less expensive and less cumbersome, so that we can develop allogeneic products ourselves, and we want to explore potential indications beyond cancer. So there's areas in either autoimmune disease or in solid organ transplantation where there could be a role for genetically modified T cells. Now, the bar is a lot higher for those sorts of diseases because there are drugs that can control T cell activation already, and so one has to offer a significant improvement to justify the cost around manufacturing and around the long-term follow-up of a genetically modified product. So we want to do this not only in the lab, we actually want to translate this into patients. And we want to do that ourselves, and we want to do that in collaboration with other companies. And what is really important to us is also to understand what happened to the T cells after they went into the patient, and what happened to the patient, of course, and to the tumor. So just sort of schematically, this is where we're focusing on. We're focusing on some of the manufacturing processes with allogeneic manufacturing and with the ex vivo stimulation, the vectors and the beads and so on that I mentioned to you that are part of the requirements of making T-cell therapies. We want to control toxicity by changing traffic control and cytokine release syndrome. And we want to address the potency issues by targeting the tumor microenvironment and antigen escape. And I think once we do all of those, we'll be enabled to go on to new targets and other indications. And so just sort of in closing, I wanted to leave you with some take-home points about CAR T-cells. These are living, long-lived, powerful drugs, mostly characterized by on-target activity, both for efficacy and for toxicity. They can induce durable responses in patients with advanced disease that are refractory to any other form of therapy. They're very bioactive in that they make cytokines and engage other cells. And so the toxicities associated with that can be cytokine release syndrome. And there's multiple different ways to design a CAR. There's just like there's more than one way to skin a cat, there's multiple ways of making a CAR T-cell, which makes the patent landscape a little bit complicated, but I think it also means that there's room for a lot of players. And there's new ones, I think, that will reduce toxicity and that will extend the platform to other tumors and to other indications. And I think this is going to be happening quite quickly, like in the next 2 to 3 years, we're going to start to see a lot of activity in this space. So I just wanted to thank a couple of people and a couple of groups. This really takes a huge team effort. It's everything that goes into a drug development, but that's been happening in the academic centers. And so this is the group in my lab. This is a cancer center support at MGH. We've developed a new MGH cellular immunotherapy program that includes much of the leadership, clinicians, and the protocol office. We have collaborators at the Broad and at MIT to come up with some really interesting CAR designs and use all the power that's here in Boston that no other place developing CAR T-cells has. So that's, I think, really our edge. And we're also collaborating with the DFHCC because they have a pretty significant infrastructure already for T-cell manufacturing and administration. And just to mention, I didn't bring the slides, but here at MGH, we've actually opened 9 new CAR T-cell protocols or engineered T-cell protocols. They're in various stages of activation, but there's been many patients infused already here with CD19 CAR T-cells, and we're starting our first trials with the TCR T-cells as well. So it's a rapidly exploding field, and it's going to be a really interesting ride. Thank you so much for your attention.
Enjoyed your talk. 2 questions. One, some of these patients who are relapsed are in pretty bad shape, and many of the lymphoma patients are elderly. What are your criteria for what patients enter your trial? And the second question is, some immunologic diseases are characterized by abnormal B-cell proliferation. Are you thinking about targeting those?
Yeah, so I'll go. The first question is about eligibility criteria for these. There hasn't been an age limit. In fact, the oldest CAR T-cell patient that I've been involved with infusing was 81 years old. It's more about general performance status and organ function. And the organ function criteria is mainly so that if there is cytokine release syndrome, that there is some physiologic reserve. So right now, when things are still on protocol, they tend to be the usual, like kidney function, creatinine less than 2, and echo with an EF of more than 55%. But I think as these cells become an FDA-approved therapy, there's probably going to be a loosening of the eligibility criteria, and we're going to learn from that what we really need in terms of making this widely applicable. I think the one eligibility criteria that is often in these protocols and not discussed as much is that there usually can be no history of autoimmune disease. And that's because in this T-cell activation process, we do stimulate all the T-cells coming out, and then we put co-stimulated T-cells back in. And so there's this fear that we will activate and kind of hyperactivate T-cells that already recognize self-antigens, which I think plays into your second question. So will there be a use for these T-cells for patients with abnormal B-cell proliferation? I think the answer is yes, but I suspect that those things won't be tried until the other therapies are approved and we get some experience in patients who have some autoimmune disease.
I think you suggested between the lines, would you worry much about the toxicity of combining checkpoints and CAR-T, not to mention the financial toxicity?
Well, there's certainly desire to combine checkpoints and CARs, and there's several open trials that are going to do that, particularly in solid tumors. I actually think it's fine. I mean, I suspect that there's going to be similar toxicities with checkpoints than there are with CARs than there are with checkpoints without CARs. So I don't think that it's going to be necessarily particularly worse, although it hasn't been formally tested yet. The financial toxicity is an entirely different story, and I don't really participate in those portions, but I suspect that the level of activity was going to have to be extremely high in order to justify the cost.
Marcela, I'd like to ask a question while Marty's walking up. There's a lot of experience in my era with making vaccines for various oncogenic proteins, for example, RAS. And there were certainly T-cell responses that were documented. Are people now exploring the possibility of pre-vaccination in terms of establishing a T-cell repertoire that will recognize the antigen and then giving the CAR T-cell?
That's a great question. I think it makes a lot of sense for when you're vaccinating and you're trying to get a broad antigen response. I'm not sure that we're there yet to be able to harness the new T-cell receptors that come out of that and turn it into a CAR or a TCR for an individual patient. But I think the whole field is growing so much that there's now databases, large numbers of databases of T-cell receptors that are going to be known, and their antigen specificity and their HLA restriction is going to be mapped as part of that. And so I think there's going to be libraries of new antigen receptors that one could potentially use and harness to turn them into drugs. But I think that's coming. It's just not quite. Yeah.
It's a complicated issue, but it seems like it's logical to me.
Yeah. I mean, if Kathy can find that the neoantigens are shared, that would make things a lot more palatable. So,
Marcela, thank you for a brilliant presentation. It reminds me of the fact that Carl June, about 4 years ago, was invited by Bruce to a previous Chabner colloquium. I was seated just in front of Joe Bertino, and he tapped my shoulder and said, "I bet you Bruce is going to bring that here to Mass General soon." So we're glad that you're here.
Thank you.
Could you just make a comment about some of the logistics in terms of the laboratory requirements in your clinical trial setting to be able to hand this expanding population of needy patients that is otherwise apparently pretty demanding?
Yeah, so I think right now there's 3 forms of dealing with the manufacturing process. So there's individual drug companies that are developing their own, right? So like Novartis and Juno, they're handling the manufacturing process. And So then the logistics operation for an individual hospital becomes about the pheresis and the chain of custody in terms of the product, but they're not doing the vector transduction and the expansion and all that. But it still actually requires fairly complex logistics, even just to administer the drugs. And in fact, there's going to be new FACT standards coming out about what centers need to do to be able to be accredited to administer the drug, and then eventually probably paid to administer the drug. The second is that there's a lot of new biotechnology companies that are growing up around this space. And coincident with that, there's also service companies, contract manufacturing organizations, which are dedicated now to growing T cells. So Lonza, WuXi, Aptec, they're becoming places where these biotechnology companies send their new vector or their new transgene and manufacture the product, and they become contract manufacturers. And then if they have signals, one would imagine that any individual company would build their own processing facility. And then the third, which is actually how these T cells came to be as therapies, is for academic centers to have their own manufacturing processes. There's a huge number of advantages to that. The companies and the commercial manufacturers need to have lockdown processes. And we actually don't think that the process right now is the most efficient that it could be. It was developed in a very sort of artisanal way. And now, there can be automation, there can be, micro reactors. There's a lot of things that I think have room for improvement. And I think that's really only going to happen in the academic processing facilities, but there's certainly a shortage of them. As far as I know, there's only one in Boston, and it's the CMCF that Jerry Ritz runs. And so I think there's a lot of, and it's very much in high demand. So there's a lot of room for improvement. If we bring in people from MIT who know all about process development, and people from Draper who are interested in, shrinking down to micro reactors and things like that, I think there's a huge potential benefit to improving the whole manufacturing process.